neuroimmunology is still something that is undergoing intensive research. So we still don't have much or all the details of what is going on in terms of the circuitry and the communication between the immune system and the neuronal system. But I just want you guys to take from this lecture this understanding that just like any other physiological process in the body, whether it's blood pressure, regulation of temperature, breathing, the immune system is part of this whole system in this homeostatic organism. And this relates then to, in the previous year, in the POD block, you thought of the immune system as a defense mechanism against pathogens, but you also learned that the immune system also played an important role in terms of homeostasis. And so that's what, in this lecture and in your subsequent lectures on immunology in third year, you will also come across. Just hang on. Hi, Montessa. I don't have access to the Moodle to download my lecture. Yeah. Okay, thanks. All right, so the objectives of this lecture is to introduce the neuroimmune network, to define the different networks where there is communication between the brain and control of the immune system, particularly in the so-called inflammatory reflex arc. And then to reevaluate, because traditionally, the reason why we always considered the immune system and the brain to be completely separate was because of the so-called blood-brain barrier, that any immune cells couldn't get through the blood-brain barrier to affect the brain, and that also had the same rule with regard to cytokines. Yeah, okay, thanks. Then I'll give a brief discussion about what we know are actual immune cells in the brain, namely the microglial cells, which have the same origin as macrophages, but they also perform specialized functions with regard to brain homeostasis. And then finally, I'll give a brief description of certain immunological diseases with regard to the brain. So first of all, why should we think about, or why should we consider the control of the nervous system to the immune system? What are the benefits in terms of the organism? First of all, think that, or consider that during an ongoing immune response, with the proliferation of lymphocytes, neutrophils, with the production of cytokines, antibodies, and other mediators, all of that takes a lot of energy. And so there has to be a system to divert the energy needed for these different functions away from other types of normal maintenance functions. So it's a matter of synchronizing and integrating different physiologies to try to optimize the best energy resources for the immune system to function. And that impinges then on the overall physiology of the person who's undergoing an ongoing immune response with regard to the inflammatory response, with regard to the high temperatures, the fever, and so on, to give the so-called sickness behavior, where you feel the effects of tiredness, loss of appetite. All of these play a role in as accessories to the functioning of the immune system during these actual processes. We also know, this is quite recent evidence, that when we sleep, that resets the biological clock. And a lot of research now is going on to the different physiologies in these so-called circadian rhythms. And we know that in the sleep state, there are more immune cells which circulate through the body compared to the waking state, where many of these circulating cells are then diverted to the periphery and to different organs. The other reason why we now understand the importance of the brain controlling the immune system is because the decisions made as a result of brain activity is a lot faster in terms of milliseconds through the passage of the neuropeptides and the neurotransmitters signaling, compared to if the immune system was a separate entity where the results of ligands interacting with receptors, and then the induction of various functions like cytokine production, cytotoxic T cell activity, so on, happens within minutes or days. The evidence that we have for this communication between the brain and the immune system and vice versa comes from various observations. First of all, if we stimulate parts of the brain or delete parts of the brain as a result of injury, this can cause a change in immune reactivity, such as cytokine expression, antibody production, or leukocyte proliferation. It's also known that in the mental state of a person, where someone undergoing stress due to emotional or environmental stimuli, this can lead to atrophy of the primary and lymphoids and secondary lymphoid organs. And the reason, or the mechanism behind this, is that we now know that these lymphoid organs are innervated with sympathetic nerve fibers. And lymphoid tissue itself is affected by neuropeptides and neurotransmitter signals coming from these connections. We also know that neurons and microglial cells themselves express pattern receptors, pattern recognition receptors, and they can make cytokines like IL-1 and TNF-alpha. Immune cells themselves, like macrophages, dendritic cells, and T cells, have been shown to express cholinergic and adrenergic receptors, showing that they can respond to signals coming from the nervous system. And not only do these immune cells express receptors, but they, particularly macrophages, can also express or make neuropeptides like acetylcholine, dopamine, and catecholamines to close this loop between the immune system and the brain. So I've said why these neural immune interactions occur, or I'll give a description of how these signals work, how do these networks work, and where in the brain are these responses controlled. The best worked out system is the hypothalamus pituitary adrenal axis. So this loop is shown in this figure. And if we first focus at the bottom of this loop, where the body gets invaded either through infection or through physical trauma, where it affects activates the immune system. Those activated immune cells can produce cytokines, which can then go and interact with various neuronal cells which have receptors that can respond to these cytokines. Once those neuronal cells are activated, they, particularly in the hypothalamus, they can induce the production of neuropeptides, which then stimulate the pituitary, stimulate the pituitary to make ACTH, which then stimulates the adrenal cortex to make corticosteroids. And these have an immunomodulatory effect on the immune system in terms of immunosuppression. So in this way, what this basically does is it regulates the immune system to prevent it from overreacting and causing, say, a cytokine storm, which can cause severe damage to normal tissues or even death. As you know, pain is one of the symptoms of inflammation. So stimulation of pain receptors, namely the nociceptors, which I think you've learned about, these activation of these receptors can occur through stimulation by cytokines, engagement of PAMPs or other inflammatory mediators, which renders the receptors more sensitive to the stimuli of, say, toxins produced by invasive bacteria, or temperature, raises in temperature, physical trauma, and so on. And activation of these receptors will then cause the axon terminals to generate neuropeptides, which can again go back to regulate the strength of the immune response, either to upregulate or downregulate the immune response. A third neuroimmunological pathway involves the vagus nerve. And this is part of the parasympathetic nervous system, because when you have a ongoing inflammatory response, once that immune response has been generated to deal with the infection, you need to downregulate that immune response, but you also need to make sure that doesn't go on unchecked, because as I said, it can result in damage of normal tissues. So with the signals coming from the vagus nerve, these can affect the various physiological responses in the body to help during this ongoing immune response to control the infection. The other thing, though, is that the vagus nerve is closely associated with mucosal sites. So imagine if you have a respiratory infection with, say, flu or COVID-19, and you experience all the symptoms of the infection, such as tiredness, loss of appetite, fever, etc. This behavior is known as sickness behavior, and it's due to the inflammatory cytokines released by the immunoreactive cells, which then act on the brain to then stimulate the vagus nerve, and the efferent signals from the vagus nerve then stimulates the parasympathetic nervous system to dampen the immune response to prevent excess damage to mucosal tissue. So much for communication between the peripheral immune system and the brain. What about immune cells in the nervous system? Keeping in mind the impermeable barriers of the blood-brain barrier and the meninges, there are three discrete points of immune activity in the brain. The first is that mediated by the microglial cells, which are brain-specific monocytes which play an important role in the homeostasis of a healthy brain by phagocytosing dying or damaged neuronal cells, and also during the process of brain development. The second are immune cells coming from the periphery, which circulate through the meninges. And this is also through these newly discovered lymphatic systems. So we now, there's intense studies going on in terms of understanding whether antigens coming from the brain can get into the lymphatics and then be going into the peripheral lymphoid system to generate homing lymphocytes to go back into the brain. These immune systems, these immune cells coming from the periphery, which circulate through the meninges, are blocked from entering the brain parenchyma by the tight junctions of the underlying glia limitans. The third are the immune cells circulating through the blood vessels, which permeate through the brain parenchyma. Under normal circumstances, they're restricted to enter the brain due to the blood-brain barrier. But under inflammatory conditions of microbial infection or autoimmunity, it's clear that these circulating immune cells can pass through, and also their cytokine products can enter the brain parenchyma through the meninges, all these blood vessels, to affect brain signaling. The solidity of the blood-brain barrier is due to the specialized properties of the endothelial cells compared to those that make up the blood vessels in other parts of the body, such as tight junctions and limited number of fenestrations. Nevertheless, studies of the endothelium in the capillaries of mouse brains show that regulated transport of cytokines can occur from the blood into the brain and vice versa. In other words, cytokines which, say, microglial cells make can exit into the meninges and then get into the circulation. Studies on mice also show that endothelial cells lining the blood-brain barrier also express pattern recognition receptors like TLR4, which can respond to LPS when LPS is injected intravenously into these mice. This endotoxin can then pass through the endothelium of the blood-brain barrier into the brain parenchyma via transcytosis through the endothelial cells. When inflammatory cytokines do breach the blood-brain barrier to enter the brain parenchyma, they can have neurotoxic effects. So what happens to the brain under the influence of an inflammatory response? As mentioned earlier, the release of inflammatory cytokines like IL-1, IL-6, and TNF-alpha in the periphery can activate centers of the brain, resulting in the so-called sickness behavior. We're still not sure about the details, how activated immune cells play an important role in brain development and repair of damaged spinal nerve cells, but some evidence suggests that Th2 cells do have a role in this through the production of IL-4. So in conclusion, the role of the adaptive cellular immune response, as brought about by T helper cells, can either be neurodestructive due to the production of various inflammatory cytokines, or neuroprotective through the activity of Th2 cells. Regarding the immune mediators involved in neurodegenerative diseases, as in the case of chronic diseases like Alzheimer's disease and amyloid, which are neurodegenerative diseases, both seem to be mediated also by inflammatory cytokines produced by microglial cells to remove or destroy misfolded proteins, as in the case of Alzheimer's disease. And in neuroinflammatory diseases, there's additional immune damage through the added invasion of inflammatory cells from the periphery, which can breach the blood-brain barrier and damage myelin sheaths. Regarding the microglia, in their normal homeostatic functions, they play an important role by regulating the number of neurons and removing damaged neurons. This is done through opsonization, using complement components, and then removal by phagocytosis. The microglial cells also play a protective role in the brain. They respond to microbial invasion by producing cytokines after recognition of the pumps, and they migrate to areas of the brain where there is damaged tissue. So based on the current concepts in neuroimmunology, a number of strategies are being developed to see whether can modulate the immune response, still different of the nervous system. For instance, for chronic inflammatory diseases like chronic bowel disease or rheumatoid arthritis, one strategy that is being trialed is to stimulate the vagus nerve to try to activate cholinergic receptors on the inflammatory cells to induce a parasympathetic response. Electrostimulation of certain parts of the brain are also being trialed with the aim of modulating the inflammatory reflex to treat physiological disorders with a chronic inflammatory etiology. There's also been another set of interesting experiments of conditioning, where what they did was to give mice a stimulus in the form of glucose tablets, with at the same time an immunosuppressive drug like cyclosporin. And they gave mice regular doses of these two chemicals. And then after a certain period of time, they just gave the mice the stimulating chemical, the glucose, and found that the mice developed immunosuppressive activity which normally was induced by the cyclosporin. So this seems pretty crazy, but experiments are underway to see whether you can condition the immune system through these sensory inputs in this way. So in conclusion, we now have a consensus that the immune system and the nervous system are in close communication with each other through several networks such as the HPA axis, nociceptors, and the vagus nerve. These alter the physiological state of the body in response to stress or microbial or pathological insult, to display sickness behavior, as well as to regulate the immune response. The brain has a functioning innate immune system conducted by the activity of the microglial cells, which play an important role in maintaining homeostasis and neuroprotective functions in the brain parenchyma through phagocytosis and the production of cytokines. So if anyone's interested in reading more about it, there's some very recent reviews about this topic. And as I said, don't worry too much about the details of this. Just try to get a general understanding of this interaction between the immune system and the brain. I haven't posted this on Moodle yet, but I will do that after this lecture. CRF, yes, that is corticotropic releasing hormone. The question, the cells that take up antigen from the brain to the peripheral lymphoid organs are the microglia? No, because the microglia do not exit as a traditional systemic, say, dendritic cells do. We don't know. We don't know the system because this lymphatic system in the brain was only discovered about 8 years ago. So still a lot of research is being done on that. We don't know. What is whether we're getting classical, just like you learned, remember, dendritic cells take up antigen, migrate to the nearest draining lymph nodes, present that to lymphocytes, which then home back to those sites. Whether this happens in the brain, it's not understood. This relation, the actual functioning of these lymphatic channels that were discovered in the brain, we still don't understand all the details regarding this. Okay, well, thanks for your attention and have a good evening.
Thank you, Doctor.
Okay.